Hello, everyone, and welcome to today's PAPRI webinar series. I'm Sarah Youssef, the PAPRI Education Working Group Coordinator and today's moderator. Thank you all for joining us to this session entitled Accessing Genetic Data in the GWAS Catalog. It's a great delight to introduce our esteemed speaker, Dr. Elliot Sollis. Dr. Sollis studied genetic and human biology at the University of Western Australia, and then he had his PhD in the genetics of language disorders at the Max Planck Institute in Netherlands. Since 2018, Dr. Sollis has worked as a scientific curator for the GWAS Catalog, based on the European Bioinformatics Institute in Cambridge, UK. His work involved gathering and interpreting research articles from across the field of human genome-wide association studies, extracting, managing, and carefully annotating this data from the publication. He also helped researchers to access the data and submit their own results. So his contribution to this field is one of a kind. In today's talk, Dr. Elliot will share his insights on accessing the genetic data in the Genome-Wide Association Catalog. So please join me in welcoming Dr. Elliot, and please be noted that we will have a live question and answer session after he finishes his talk, and that the webinar is recorded and will be uploaded to our YouTube channel. So I ask you to kindly mute your microphone so Dr. Elliot can start his talk. The floor is yours, Dr. Elliot.
Thank you very much, Sarah. Thank you for the very kind introduction, and thank you for inviting me to speak today. As you mentioned, I'm one of the curators at the GWAS Catalog, and I just wanted to mention that there is a team of us, so it's very much a group effort. And today I'm going to talk to you about accessing genetic data in the GWAS Catalog. So I'm going to start by giving an overview of some of the key concepts in GWAS. Many of you may be very familiar already, some might not be, so it's good to just recap some of those concepts. I'm going to give an overview of the data and metadata that we have available in the GWAS catalog. I'm going to show how you can search the catalog to find data that may be of interest to you. And I'm going to touch on how you can access full GWAS summary statistics, which I'll explain in a little bit more detail later on. So beginning with some key concepts. Genome-wide association studies are a well-established method for identifying associations between variants and traits across the genome. So you would typically look at, for example, cases who have a disease or other trait and compare them to controls who don't have that trait. And then you're looking at all of the different points of variation across the genome.
Yes.
Sorry? Okay, I think I'll continue. You're looking at all of the different points of variation across the genome and looking for places where a variant is in higher frequency in the cases than in the controls. And the strength of each of those associations is indicated by a p-value. And GWAS can be very valuable for understanding common and polygenic traits in particular, including a number of diseases. And it has lots of downstream applications that make it very useful for other types of studies. So a typical GWAS would involve first defining a sample of individuals from a population. For example, you may have cases who have asthma and controls who don't have the disease. You would genotype variants across the genome and find alleles that are significantly higher frequency in the cases than in the controls. So for example, if 58% of all of the asthma cases had this A allele at a certain position, but only 17% of the controls had that A allele, then you would conclude that this A allele is associated with asthma, with an increased risk of asthma. And that would be considered a significant finding if it were at a p-value below a certain threshold. So normally 5 times 10 to the minus 8 is a typical genome-wide significance level that many researchers use. We're a little bit less stringent about that when we look at which variants we will include in the GWAS catalog. It's just to give people as much information as possible, and they can make their own decisions about which ones they think are most likely to be important. So there are also some different kinds of GWAS that can be conducted, and this is partly based on whether the trait that's being looked at is a binary trait, so a case control kind of study, or if it's a continuous quantitative measurement. So I already talked through the example of a case control study on asthma, where you're looking for an allele that's associated with an increased risk of getting asthma. But on the other hand, you can have quantitative measurements where, for example, you might be measuring cholesterol levels in a population, and you'd be looking for alleles like the A allele here that are associated with a higher average level of that particular molecule. So here you would say that the A allele is associated with increased risk, increased cholesterol levels. GWAS often have two different stages as well, a discovery and a replication. So for example, in our asthma study, they might have analyzed more than 100,000 variants or even millions of variants, but then narrowed it down to just 5 that reached the significance threshold. And then they might take those 5 variants and test those again in a separate sample of new cases and controls, and use either GWAS again or a different, more targeted method to determine whether those 5 significant variants remain significant in the replication stage. So they may either pass or fail validation. So the output of a GWAS study takes the form of summary statistics, which is basically a large table of all of the results for all of the variants that were analyzed. So it could have 100,000 to even millions of rows, and it will have information like the location of the variant on the chromosome, the alleles, the effect allele that we're actually looking at that has an effect, and the other allele, as well as the effect allele frequency. We would also have information about the size of the effect and some sort of measurement of the error or the range in that estimate, and a p-value to indicate how significant the result is. But often in a GWAS publication, the ones that will really be talked about are just the most significant associations. So just those that have a very small, very significant p-value. So you might see something like 3 significant loci were identified in the paper. And a final important thing to keep in mind about GWAS is that an associated variant is not necessarily the causal variant. So if a variant is associated with asthma, it doesn't necessarily mean that that's the variant that causes an increased risk. And that's because of this idea of linkage disequilibrium, where 2 variants that are located close to each other in the chromosome can tend to be inherited together. And so you may see that many people with asthma have this particular SNP that we've genotyped, but actually that's not really responsible for the disease. There's actually a risk SNP that's somewhere nearby that's being sort of carried along. So it's always important to do further study to understand what an association actually means. So in the GWAS catalog, we have lots of different data about GWAS studies. So firstly, we have information about the publications, the the scientific journal articles that report the GWAS studies. And at the moment in the catalog, we have nearly 7,000 different publications represented. But each publication might have multiple different GWAS experiments in it. So they might be looking at lots of different types of a disease, or they might be looking at multiple protein measurements or something like that. So you can get up to a couple of thousand different GWAS in a single publication sometimes. So at the moment, we have more than 113,000 individual studies in the catalog. And for wherever possible, we host the full summary statistics for each of these studies. It just depends a little bit on whether the authors choose to share them or whether we have the right permissions to reshare them with other people. So we actually have a very high percentage of studies at the moment that do have summary statistics. So we have 97,000 of them at the moment. And then in addition to those full data sets, we also extract the top associations from the papers. So they have to have a p-value of less than 1 × 10⁻⁵. And at the moment, we have about 670,000 individual associations. So where does this data come from? In theory, we try to include information about all of the GWAS that are published that are conducted in humans. And they come from 2 sources. So we curate published papers internally in our team. And we also receive direct submissions of data by authors. So both of these come together to create the GWAS Catalog. We have a lot of information about the studies. So one of the first things is information about the trait that was studied. So we report the trait in 2 different ways. We have something that we call the reported trait, which is a free text description of the trait. And this is supposed to match the author's language as closely as possible. And it can be quite specific, quite detailed. And the idea here is to provide as much information as possible to users about exactly what was studied in that particular study. So for example, we might have a trait called asthma, moderate or severe, where they're just looking at those more severe cases. And then on the other hand, we have terms that we've taken from the experimental factor ontology, which is a hierarchical, very standardized list of terms. And we use 1 or more terms from that ontology to annotate each study. And the idea of these is they should be consistent across similar studies. So we sometimes can omit those very specific details in the interest of making it clear that all of these studies are on something very similar. And it's the ontology terms that we use to drive the searching of our database online. So an example here would be, we would summarize this as simply asthma in this case. And it has a standardized identifier to make sure you know you're always looking at the same thing. So the experimental factor ontology, as I said, is a set of standard terms that cover many different aspects of biological research. And these terms can represent nearly all of the traits that might potentially be studied by GWAS. And we're always adding new terms to the ontology to reflect changes in the kinds of things that people are studying. And the real benefit of it is that it's arranged in this hierarchical structure where you have child terms, like these very specific subtypes of asthma, which are then grouped together under the parent term of asthma, which itself is a child of another more broad term, and so on. So you can sort of choose how granular you want to go when you're looking for data in the catalog. We also record information about the sample size, so how many people were studied in the GWAS, and also their ancestry. So that's particularly important because you want to make sure that if you're comparing two studies, that you know how similar the genetic background is, or whether it differs, because that can affect the interpretation of the results. And we capture the ancestry in two different forms. We have a free text sample description, which again reflects more how the author describes it. And then we also have a more structured sample description where we use controlled terms to try to increase the consistency and improve the search functionality. And we have a paper here where we go into much more detail about how this standard structured description was developed. So, for example, I have two different studies here with the free text description at the top and the structured description at the bottom. One difference to note is that the structured description also includes a list of all the countries that the study participants were recruited from. And we also may have a more specific population descriptors in the free text, for example, Japanese ancestry. And in the structured one, it's grouped only into these kind of broad continent level ancestry categories, in this case, East Asian. And also where we have cases and controls separated out in the free text, those are added together in the structured sample description, just to give a total number of how kind of big the study is. So the next thing that is annotated in the catalog is information about the genotyping technology. So the kinds of arrays or sequencing methods that we use to do the genotyping, as well as the total number of SNPs that were analyzed in the study. And then we have data about the top associations and the full summary statistics. So when it comes to the top associations, I think I've said this a couple of times already, that we extract just the most significant associations. And we have just one association for each independent locus. And it has to be significant in all of the stages that were included. So if there was a discovery and a replication stage, then we only extract the SNPs that are the associations that are significant in both stages. So the information that we extract for the associations is information about the variant. So the variant identifier, which is an RSID, the chromosome and base pair location, and the nearest gene. We extract information about the alleles, the effect allele and the effect allele frequency, and information about the significance of the association with the p-value, and finally the effect size. So if it's a case control study, this would be an odds ratio, and if it's a quantitative study, it would be a beta. So yeah, the last thing is the full summary statistics, and I'll talk about that in more detail in the next section. So now I'm going to talk about how to search the GWAS catalog and find information. So this is what the homepage looks like. And I had the link here. I'll share the slides with you afterwards, I think. So you should be able to access any of the links that I have in the slides. So at the top, there's a search bar where you can search, for example, for variants, genes, traits, or specific publications.
Yes, Olunga. Ayo, jatoka wende nyumbani.
There's a download section where you can download the GWAS catalog data as a spreadsheet. There is also a summary statistics page where you can go straight to the studies that have full summary statistics. And you can also submit your own summary statistics if you have done a GWAS yourself. And we have lots of documentation with additional information about how we prepare the data and other ways to access it, like using an API to access programmatically. So just looking at the search function at the moment, one common thing to search by might be if you have a specific publication in mind. So maybe you're interested in this particular paper. The best way to search for a specific paper is to use the PubMed ID, which is a unique identifier for each publication. So you can search for the PubMed ID, and this will give a list of, well, it should just be one matching result. And so if you click on the search result, that will then take you to the publication page. So this has lots of information about the publication. So for example, all of the citation details, but also links to read the paper in PubMed or Europe PMC. And then we have a summary of all the available data for this publication. So all of the associations that were reported in here, if there were multiple different studies, they'll be listed. And also if any of those studies have full summary statistics available from the catalog. So if we just look at the studies results, so this gives a list of all of the different studies in that paper. So in this one, we have just two. It has information about the paper itself here, about the traits that were studied in each study, information about the samples at the discovery and replication stage. So this one is just a discovery only publication. And then we have a list of how many associations, how many significant associations were extracted from the paper for each of these studies. And then if there are full summary statistics available, we have a link here to where you can download them. One thing that you can do on this table as well is to use these little search bars to filter your results. So say you are particularly interested in studies that had individuals of African ancestry, you could search the word African here and it would filter the table to just show the ones that match. So here we have a number of samples of African ancestry here. In this case, there are only two, so you can see quite easily. But sometimes there are many, many studies, hundreds or thousands within a publication, and it can be useful to filter here for just what you're looking for. There's also these links here under the study accession table. So this is a unique identifier just for each individual GWAS in that publication. So if you click on that, it'll take you to a separate studies table, a studies page, sorry, where there's a lot more detailed information about the study. So here at the top, we have information about the trait, about the sample descriptions, and lots of other things. It also tells you if there are full summary statistics available. And it tells you if, it tells you which license this data falls under. So typically, all of our data is free to use for any purpose, but there are occasionally some differences, so it's worth checking the license information here. So on the study page, there's also an associations table, which lists all of the significant associations that we extracted for that study. It has information about the variant and the allele, as well as the p-value, the allele frequency, the effect size data, and the nearest gene. And then it has information about the trait, the study accession, so you can link back to the study page, and the location of the variant on the chromosome. Probably the most common search method that people would use is to search for a trait of interest. So if you're interested in chronic obstructive pulmonary disease, you can search for that. and then click through to the trait page for that particular disease. And this will give you now all of the data in the catalog that has anything to do with this disease. So we have the information about this trait at the top, where you can find it in the ontology. Some synonyms, often there are multiple different names for a particular trait, and a description. And here you can link to look at that trait in other places. So there's the top two ontology sites where you can look at how that trait relates to other traits. You can also look at it in open targets if you're interested in drug discovery. And probably the most important, most useful thing for this audience is the links to the GWAS Catalog, where you can find information about polygenic scores for this same trait. Something that's worth looking at here is the reported traits. So this shows all of the more specific reported traits for this disease. So lots of different versions of chronic obstructive pulmonary disease with slightly different nuances. And there's also a child traits button where you can look at the more specific terms that come below this in the ontology. So in this case, these are subtypes of chronic pulmonary disease that fall under that same overarching category, but they're different, more specific diseases. And here we have a summary of all of the available data for this disease. And one thing to notice here is this include child trait data. So if you, for some reason, were not interested in any of these specific subtypes, but just wanted to look at studies that were on chronic obstructive pulmonary disease as it is, you can uncheck that box and it will remove some of the studies from view. So you'll get a slightly trimmed down set of data. Another thing that you can look at on the trait page is a visualization of all of the associations for this trait. And this is plotted here with all of the chromosomes along the bottom. And this is the p-value on this axis. So the most significant ones will be at the top. And you can also click on these to get more information about exactly how significant they are and which study they come from. You can also download this image from the site. So other things you might want to search by are variants, genes, or regions. So these are what the search results will look like for those. I'm just going to highlight the variant page at the moment. So say you're interested in this particular variant, it's in this gene, and we have some other information about the location. We have information about the alleles, and again, links to information about this variant in other resources. And here we have, again, the same summary of data, where you can see all the associations with this variant. You can see all the studies that reported it. And something new here is the trait button. So here you can look at all the different traits that have associations with this variant. This is often quite an interesting view to look at because here, for example, you can see that all of the traits that are associated with this variant have something to do with blood pressure. So you can start to see patterns emerging. And you can see here an association count. So how many times has this variant been associated with systolic blood pressure, for example? Then you can also rank these to see which ones are sort of the most frequently reproduced results. Here we have the linkage disequilibrium plot. So I mentioned earlier the concept of linkage disequilibrium. So this plot will show the region around the variant that you're looking at the moment. And it will show other variants that may be in linkage disequilibrium with it. So that means basically other variants that could be tagging the same association signal. So the ones that are in closest LD are up the top of this graph. And you can click on the others that are nearby and see which variants they are, how closely they are linked, but also you can see the other traits that that's been associated with, and see, for example, in this case, that they are also associated with some very similar traits. You can download this data as an image or as a file that you can do more analysis on. But another useful thing about this is that you can change the reference population. So at the moment, this is set to a British population from England and Scotland. But changing the population often changes the LV scores. So if you're looking at a different population, you might want to see the LD for that population, which might be more relevant to your interpretation of your own findings. So, for example, if I change this to a Yoruba population from Nigeria, then that variant that we looked at before is no longer quite so closely linked. The LD measurement is not as high. So in this population, you might make some different conclusions about those neighboring variants. So you can also, as well as searching the website, you can download the catalog as a huge spreadsheet and do your own analysis on it. So again, from the home page, you can click on the download button and go to the files page. And here you can download a list of all the associations, which is a very big file, a list of all of the studies, if you just want to see which studies have been done on which traits, for example. And you can also download the ancestry information across all of the studies. There's a link here on the right that just goes into some detail about what all of the different columns in these files means. And this is just a quick example of what that spreadsheet looks like. So each row, in this case, this is the associations download. So each row is a different association. And then you have information about the publications that they came from, the traits, and other details about the variant. So the other thing that you might want to do is access the full summary statistics for a particular study. So again, that's those large tables of all of the variants that were analyzed. And full summary statistics have a lot of uses. You can use them to combine different studies and do GWAS meta-analyses. They're also one of the main things you need to run a polygenic risk score analysis, polygenic score, which might be the most interesting for this audience again. There are also other analyses you can do, like Mendelian randomizations, looking at genetic correlations between different traits, estimating SNP heritability, and also combining the GWAS with other omics data, like proteomics or metabolomics, for example. To access full summary statistics, you can follow the summary statistics section of the homepage, which can take you to a list of all of the available studies. And this is broken down, firstly, all of the published studies. So these are ones that are associated with a published journal article. And here you can see all of the information about each study and then use these links on the right to download the summary statistics. And then we also have a separate table for unpublished studies. So these are ones that are not yet in a journal, but the authors have already shared them with us and already said that they can be released publicly. So this is often the newest, most kind of the most recent research. And this again has information about all of the studies. One thing to be slightly cautious about is that this has not necessarily been reviewed by a curator yet. So it's just exactly how the authors have provided it. That's still very useful information. And again, you can follow these links to download the summary statistics from our FTP. You can also get to the summary statistics through a specific search, like I showed before. So if you were interested in hepatitis C virus infection, you could go to that page and then click on the full summary statistics button to see a table of all the studies on that particular trait that have full summary statistics. And again, it has all the information about each study and then the links to download the summary statistics. So once you follow those links, this is what the file structure looks like on the FTP. So we have all of the different studies are identified by their GCST identifier, and they're grouped together on the file system in groups of 1,000. So you have this folder here, and then each individual study has its own subfolder named according to the GCST identifier, like that. And then within these folders, you'll see a main summary statistics file, which starts with the GCST. It has the genome assembly, and it usually ends with .tsv or .tsv.gz. There's also a metadata file, which gives you more information about the study. And in a lot of cases, but not necessarily all of them, there'll be a subfolder for the harmonized summary statistics, which I'll talk about in a moment. So the summary statistics themselves, if they're hosted on our site, we require them to be submitted in a specific format. So the current format is called GWAS SSF, and that's been our standard format since April last year. And they have a specific set of mandatory fields that have to come in a specific order, and then another set of fields that we encourage people to add, but they're not mandatory, which come after that. And then users can also add as many additional fields as they want to fully describe how their study was conducted. If you're interested in the full specifications, I have a link here to the specification document. Then this is the metadata YAML file. So this shows you some information about the file. It has basic information about the file, how the genotyping was done, information about the samples, and about the traits. One thing to note is that we have a couple of different file types. So the current standard, anything that meets our current requirements would have the file type GWAS SSF version 1.0, and we'll increment this as changes are made to the standard over time. Before we brought this in, we were already accepting submissions. So there's an earlier pre-GWAS SSF format. So these are often still very useful files, but they don't have, didn't have quite the same requirements. So they may be missing some fields. And then others are non-GWAS SSF. So these are ones that they're a slightly different kind of GWAS and they just don't really fit into our standard format. So that's if people have done studies on gene-based association studies. So where you're looking at whole genes rather than individual variants or copy number variation. So we still host those, but they're done a little bit differently. So lastly, we have the harmonized summary statistics. So the idea of these is to maximize the interoperability of the data sets to make sure you can merge them together and use them and compare them. We make the summary statistics available in this harmonized format wherever we can. And so these have their own subfolder, and they'll have, again, the file in the folder. And the things that we do with the harmonization is that we map them all to the same reference genome. So currently the reference genome is GRCH38. We also make sure that they're all described with the alleles reported on the same strand, and all of the variants are sorted by chromosome and base pair position. So finally, if you have your own summary statistics that you would like to submit to the GWAS catalog, or if maybe this hasn't occurred to you, here are some of the reasons why this can be very beneficial to the sharer. So sharing summary statistics can help to enable collaboration. It puts your data out there so other people can use it. Sometimes that simply just fulfills the requirements that you need to submit your work to a journal. But there's also good evidence that when you do share your data, you get more recognition for the work and get increased citations compared to people who don't share. It also just enables reproducibility, means that people can run analyses on your work and builds trust in the results, and also encourages other people to share their data as well. So we take submissions of summary statistics for GWAS that are already published in a journal. So no matter how long ago the work was done, we can still accept new submissions of summary statistics after the work has been published. But we also accept submissions before the publication has appeared in a journal. And so sometimes journals require you to do that before they accept the publication. But also it's just a good way to get the data out there as quickly as possible. And yeah, so they all have to meet our standard GWAS SSF format. And then the results are available to download from the GWAS catalog, together with all of this supporting information that I've talked about. If you're interested in submitting your own work, we have another webinar online that you can follow. And we have lots of very detailed submission instructions on our website as well. We also have some other training materials available on our related resources page, lots of different links to online tutorials and webinars that you can watch. And lastly, I've just got some places where you can get further information. We have a frequently asked questions page on our website. We also have 2 mailing lists. We have GWAS subs, where you can get help with your own data submissions if you're in the process of submitting summary statistics, and the GWAS info email address, where you can ask any general questions you have about the data in the catalog, how to access things, or anything that you don't understand or that you think might be missing. So that's everything I had today. Happy to take questions.
Yeah, thank you very much, Dr. Sollis, for this very detailed presentation. So for everyone, you can use the raise your hand button, or you can just type your questions in the chat box.
I'll just open the chat as well so I can see. If there was anything I went over too quickly as well, I'm happy to go over things again.
Yeah, sure, Dr. Hisham, you can ask a question. Go on. Just unmute your microphone and go on.
Thank you. Thank you very much. Thank you for the interesting presentation. I am Hisham, studying at Nagasaki University in Japan. Is there too much difference between human GWAS and microbial GWAS, especially in terms of drug resistance? Because when I accept, I think this the GWAS catalog is only for human studies, not related to microbial studies, for example, drug resistance and these issues.
Yeah, okay, yeah, that's true. Yeah, so we are just a resource for human GWAS studies. I guess there is a connection with microbial research as well, because we have studies on the human host's reaction to infection. So if you're interested in a particular infectious disease, we do have information about differences among humans that influence how susceptible they are to getting an infection. But when it comes to the microbial genome itself, I am not an expert in that. I don't know very much about the microbial genome. I imagine it is quite different, quite a different. There are lots of details that are very different, but yeah, there may be resources out there as well for microbial genomics that use a GWAS or a similar method, but I'm not sure of what those are.
Thank you very much. Thank you
So
again
the
for your
question, Dr. Oludayo, you can go on with your question, and then Dr. Mona can go afterwards.
Okay, thank you so much. My name is Oludayo Akinshola from University of Jos, Nigeria. Just for clarification.
Yeah.
Hello, Doctor.
Can you hear me? Just for clarification, like in animals, there's a way we detect selection signatures or selective sweeps.
Okay.
Is it similar in humans? That's just a clarification I want, because I know that there are different models in detecting selective sweeps, positive selection signatures. Do we do the same thing with human genome?
I'm not sure actually. I know that, well, so on one hand, I know that there are also many GWAS conducted in animal species and also agricultural plants. So some of the same methods can be applied in that direction. I don't know much about selective sweeps myself, so I'm not sure, and it doesn't really come up that I can remember often in human GWAS publications, but there might be a separate body of literature that deals with that potentially.
Okay, thank you.
Okay.
Yeah, Dr. Mona, please go on.
Thank you a lot for your talk. Can you clarify, please, a little bit on the linkage disequilibrium and how can we use it on the GWAS catalog?
Yeah, sure. I'll just go back a little bit. Hang on. Okay, I'll go back to the linkage disequilibrium plot. So this one. So basically, the idea is that if two variants are very close together on the chromosome, then they tend to stay together when they are passed from one generation to the next. So that means that people who have one variant, have a particular allele for one variant, are very likely to also have a particular allele for the variant next door. So even though they may do very different things biologically, there is a greater than chance probability that you'll have, if you have A for this variant, you'll probably have G for this variant, for example. And then people who have G for this one might have C for this one. And that's just a kind of a statistical tendency. So that means that if you have found an association signal between this variant and a particular trait, you don't know if this variant is actually causing that trait, or if it's actually one of the other variants nearby that are in linkage disequilibrium with it that might actually be the more biologically significant variant that might actually be causing that disease, for example, or contributing to the risk of that disease. So that just means that you have to be a little bit careful with the association that you find, that there actually is probably a range of different SNPs that are nearby that could be also involved. So this LD plot just kind of allows you to visualize that block of linked variants. So here for this variant, we can look at this plot and see what else is nearby that's kind of linked together with it. So all of these ones that have a very, very high R² value means that they're in very close linkage disequilibrium. So if you found a signal with this one, it's probably the same as if you found a signal with this one. So basically, that's kind of what this is for. But these linkage disequilibrium patterns do vary quite a lot between populations. So that's why it's useful to be able to look at these. I'm not sure that this is kind of something that every user needs necessarily. So it may be more if you have some more specific question you're trying to answer that's related to linkage disequilibrium, or if you are trying to really narrow it down to find the causal variant, which is kind of a whole other kind of analysis that you can do. But this would provide you some of the information you might need to do those analyses.
Yeah, great explanation. Thank you. So your cutoffs, the cutoffs you use for the squared values are like 0.8. You're using 0.8 here, or what are the cutoff value that are?
Yeah. So here there isn't. Okay. Yeah. So at the moment, no cutoff is selected. So it's showing everything. So it's showing things with high R² and things with low R². But there is this button here where you can filter it. So I think what happens is if you click this and it's set to 0.8, then anything below 0.8 will just be kind of removed. So you don't have to look at it. But it is, it's quite flexible. You can do whatever you want with it. You can set your own cutoff, and you can also change from R² to, I forget what the other measurement of LD is. I think it might be D. Can't remember, but you can change it, and you can change how wide the plot is as well. How much of the chromosome you want to see.
So what's usually the significant cutoff that above it there's a linkage disequilibrium, and below it's there's no.
I think it probably varies a little bit. I'm not totally sure if they're the factors that you would need to take into account to do a really good analysis. But I do think that 0.8 is very widely used. And at least I mentioned that when we extract the top associations from the papers, we try to only take one variant per locus. And so when we have lots of different variants that are close together that are reported by an author, we do often use 0.8 as a rule of thumb to kind of only take the most significant ones, and anything that's linked to it that has a less significant p-value we can leave out. So 0.8 is what we often use, at least.
Okay, thank you. It's a great plot. Thank you very much for explaining.
Thank you.
Please, can I ask a question here? I'm sorry. For the how the plot, do we need to use a pruned data set for the genotypic data? Is it a standard to prune our data set or not?
I'm not quite sure what you mean. The populations themselves here that you can choose from are standard. They're from 1,000 genomes, I think. Is that what you're thinking of?
Okay. I'm fine.
Okay. Great.
Okay. Thank you.
Okay, so Dr. William is asking in the chat if someone can use the available genetic data to perform a Q2L analysis of some traits.
Possibly. Okay, sorry. So there are two questions, I guess. Are there crop genetic data in this GWAS catalog? The answer to that one is no. This is human only.
Answered, yeah.
Yeah. The other one, can someone use this to perform a QTL analysis of some trait? I'm not sure. I'm not an expert in doing QTL analyses. If you do know what you need to conduct one. I could tell you whether we have that data or not, but I'm not sure exactly.
Okay, anyone has any more questions, or shall I just conclude?
If there was anything I wasn't able to answer today, please feel free to send us an email as well to the GWAS info email address that I had on the slides, and we can always look into things in more detail if you have other questions.
Yeah, perfect. Thank you very much, Dr. Sollis, for answering the questions. So by that we conclude our session for today, and I would kindly ask the participants to fill in the survey. It will pop up in your window after the meeting is finished. So thank you for having you all today, and have a great day. Thank you, Dr. Sollis, for the nice presentation, and thanks for having you.
Thank you very much. Thanks for all the great questions.
Thank you, Dr. Sollis.
Thank you.
Bye.
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 Yeah.
Thank you. Bye-bye.